When you hear the word virus, what's the first thing that pops into your head? Probably getting sick, right? But what if I told you that some viruses are not only harmless to us, but are actually one of the most powerful tools in modern science? Yeah, it's a wild paradox, and we're going to unpack it. Ah, yes, this guy. We've all seen this image, haven't we? For a few years, it basically became the symbol of a global threat. And for good reason. Viruses are ridiculously good at what they do. But what is it that they actually do? Okay, so at their absolute core, viruses are, well, they're biological pirates. The definition here nails it. Obligate intracellular parasites. That's just a fancy way of saying they are completely helpless on their own. They can't eat, they can't grow, and they definitely can't make more of themselves. To do anything, they have to hijack a living cell. So how exactly does this tiny hijacker pull it off? Well, it all comes down to its design, which is it's brutally elegant. It's like a masterpiece of minimalist engineering, built for one thing and one thing only. All right, first, let's get some lingo down. When a virus is fully assembled and ready to infect, we call that a virion. You can think of it as the virus in its little travel pod. It's basically just a set of genetic instructions. It's DNA or RNA tucked inside a protective shell. And that protective shell has a couple of key parts. The main structure is called the capsid. It's this tough protein coat. But some viruses, the really sneaky ones, add an extra layer of camouflage. It's called an envelope, and they literally steal it from the host cell as they're busting out. It's exactly like a thief running out of your house wearing your jacket. Just incredible. And this diagram, it shows you exactly how all those pieces fit together. See? You've got the genetic code, the genome, right on the inside, that's wrapped up in its protein capsid. Put those two together, and you get the nucleocapsid. And for a virus like this one, the whole thing is then smuggled inside that stolen envelope, which is studded with these little proteins that act like keys to unlock the next cell. And believe me, they do not all look the same. Nature has gone wild with viral designs. Some look like a 20-sided die, that's icosahedral. Others are long and spiral-shaped, called helical. And then you get the complex ones, which look like they flew right out of a sci-fi movie. But no matter what they look like, pretty much all viruses follow the same 5-step master plan to take over a cell. Think of it like a heist. Step 1, attachment. They latch onto the right target. Step 2, penetration. They break in and inject their blueprint. Step 3, synthesis. They force the cell to start manufacturing viral parts. Step 4, assembly. All those new parts start putting themselves together. And finally, step 5, release. The new crew of viruses bursts out, ready to hit the next target. This image just brings that whole 5-step heist to life, doesn't it? Here, we're seeing a very specific kind of virus. It's called a bacteriophage, and you can see it landing on a bacterium, injecting its DNA, and just turning that cell into a non-stop virus factory until, boom, it bursts. It is a complete and total hostile takeover. Now, that virus we just saw, the one attacking the bacterium, that's the real hero of our story today. See, while we're usually worried about viruses that infect us, there's this whole other universe of viruses that have a completely different target. So let's meet the bacteriophage. This right here is a T4 bacteriophage, and if your first thought was, hey, that looks like a lunar lander, you are spot on. That's what everyone thinks. It's basically a microscopic machine designed to do one thing: land on bacteria and inject its payload. Okay, so here's the absolute most important thing to know about bacteriophages, or phages for short. They are total specialists. They are physically designed to only infect bacteria. They completely ignore human cells, animal cells, plant cells, you name it. Their keys only fit the locks on bacteria. And you know what they say, the enemy of my enemy is my friend. All right, so once a phage gets its DNA inside a bacterium, it faces a big decision. It's a strategic choice. Does it go for a quick, violent win, or does it play the long game and go into stealth mode? And those two strategies are called the lytic and lysogenic cycles. The lytic cycle, that's the all-out assault we saw before. The virus makes a ton of copies of itself and then literally blows the cell apart to get out. But the lysogenic cycle? Oh, that's way sneakier. In this one, the phage DNA just slides right into the host's own DNA and becomes a silent passenger. It gets copied every single time the bacterium divides, spreading itself without anyone even knowing it's there. This slide gives us a perfect map of these two pathways. Up top, you've got the lytic cycle. Bam, get in, get out, destroy everything. On the bottom path, you see the lysogenic cycle, where the phage DNA just chills out, incorporated into the host genome. But look closely. See that little arrow? Under the right conditions, like when the host cell is stressed, that hiding phage can flip a switch and jump right into the destructive lytic cycle. It's a calculated move. Okay, so why are we talking so much about bacterial viruses? What's the big deal? Well, here's the twist. Scientists looked at this tiny, specialized hijacker and didn't see a threat at all. They saw an opportunity, an incredible one. They realized they could turn this natural-born killer into a precision tool for the lab. Everybody, meet the M13 bacteriophage. Now, this one doesn't look like a lunar lander. It's filamentous, meaning it's long and thin, kind of like a piece of spaghetti. And this little guy has become an absolute workhorse, a superstar, in molecular biology labs all over the globe. So what's so great about M13? Why is it the perfect tool for the job? Well, it has a few killer features on its resume. First, its genome is single-stranded DNA, which is perfect for a key technique called sequencing. Second, it has a non-lytic life cycle. It doesn't kill its host. It just secretes new virus particles, which means scientists can have a continuous, stable supply. It's like having a factory that never shuts down. Plus, it can carry surprisingly big chunks of extra DNA, and its favorite target, E. coli, is the most common, easy-to-grow bacteria in any lab. It's a perfect match. So because of all these amazing features, scientists had a brilliant idea. They figured out they could hack the M13's genetic code, basically turning it into a tiny, biological FedEx truck to carry and deliver any piece of DNA they wanted to study. So here's how it works in a nutshell. First, you get the phage's circular DNA by itself. Then you use these things called restriction enzymes, they're like molecular scissors, to snip it open at a very specific spot. Next, you paste in the gene you're interested in, you seal it all back up, and then you just pop this newly engineered DNA back into some E. coli. The bacteria and the phage do all the hard work for you, making millions and millions of copies of your gene. And this isn't just theory, this is the real deal. This is a map of an actual engineered M13 vector that scientists use every day. You can see where they've cleverly added new things like this polylinker site. That's just a stretch of DNA they designed with tons of different places for those molecular scissors to cut. It makes it super easy to slot in new genes. It's an absolute masterpiece of genetic engineering.
And of course, M13 isn't the only viral tool in the scientist's toolkit. There's another big one you should know about, bacteriophage lambda. It's also a powerhouse for cloning, used to carry and copy foreign DNA.
And here it is, bacteriophage lambda. So it's just amazing, right? The very same type of biological machine, a virus, that we fear when it comes after our cells, becomes one of our most indispensable allies in the lab. We've taken the ultimate hijacker and put it to work for science. It really gets you thinking, doesn't it? We've managed to turn a natural predator of bacteria into a gene cloning machine. It's just such a powerful reminder that in the natural world, the line between a foe and a friend, or a threat and a tool, it's really just a matter of perspective. Makes you wonder, what other secrets are hiding in the microscopic world just waiting for us to discover them?